Well, thank you to all of you for joining us today. It's a real pleasure to spend some time together to talk about this really important topic. So indeed, the title of the talk today will be Predictive Preclinical Oncology Studies Using Patient-Derived Xenograft Platforms. Um, but before we jump into the technical content for the day, I'd like to give you just a little bit of an overview of who we are at the Jackson Laboratory. So our mission is to discover precise genomic solutions for disease and to empower the global biomedical community in our shared quest to improve human health. And we do this through a number of separate but very interconnected arms. So the Jackson Laboratory is, in fact, a not-for-profit biomedical research institution. And so we have independently funded investigators that are working right alongside of you on some of the most complex and pressing problems related to human genetics and disease. And so our research focus and discovery focus all interplay with our commitment to provide education, such as the content that you're joining us for today, to kind of result in this innovative model to be able to bring you the most cutting edge technologies and services in regard to these research challenges and questions. So where are we headed today? There are a few different points that I'd like to highlight for you. In the first section, what I'd like to first do is describe to you how NSG mice are a useful platform for human disease research, including oncology modeling. So in all of this, it's important to first talk about the platform, starting with the mouse as the host here. And then next, what we'll do is demonstrate how PDX models are used for predictive in preclinical research. And then finally, we'll touch briefly on immunologically humanized models for immuno-oncology applications. I know there were quite a number of questions submitted about that topic in our pre-submitted questions, so thank you to those of you who sent those in. We'll have a little bit of that here, but then we also have several other talks that go into that topic in much more detail that we would be certainly glad to direct you toward. So before we jump into the content, I'd like to know just a little bit about where you are in your programs in regard to using patient-derived xenograft models in your research. So Dolores has opened a poll question for you very near your Q&A pod. So if you could please respond to that question by selecting the radio button and then clicking submit at the bottom of that panel, just to let us know a little bit about where you are in regard to PDX models. Just it'll help me have some context about who's out there and what your focuses are, and it'll help me direct the content a little bit better. So thanks so much for weighing in. Oh, there we go. Great. Awesome. Thanks, Dolores. Okay, so a little bit of a mixture here. Looks like maybe about a third of you are currently using PDX models. And many of you are here for education purposes. That's awesome. And some are in the planning phases. Great, great. Well, I think we've got a little bit for all of you. And I appreciate your feedback here. That's really helpful. All right, so let's go ahead and dive right in. And we're first going to spend some time on focusing on the platform in a broad context to kind of give you some orientation. But let's start with, the need here. So only 5% of cancer treatments that enter clinical trials are ultimately approved. And a major cause of attrition of these test articles is lack of efficacy and safety. And so one of the ways to hopefully improve these statistics here would be to employ the earlier use of more predictive preclinical models to diminish this attrition once these agents enter clinical trials. So that's really the broad context for this talk today. And historically, much of the cancer research that has been done using this classic xenograft model using nude mice. We see here an example of a nude mouse engrafted with the MCF7 breast cancer cell line. We see a easily visible and measurable tumor on board in the mouse that is then able to be treated with a drug, and you see regression of that tumor. So it's really a workhorse of a model. But the limitation that you have here, or one of the key limitations that we have here, is at the level of immunodeficiency in that nude mouse. So the nude mouse itself carries a mutation, spontaneously occurring mutation in the FOXN1 locus, FOXN1 nude mutation, that when homozygous results in this hairless phenotype, and it also impairs thymic follicular patterning, which results in a lack of functionally mature T cells, and then of course crosstalk between B lymphocytes as well. So the primary immunodeficiency in the nude mouse is at the level of the T lymphocyte and the B lymphocyte. There is intact innate immunity, intact cytokine functionality. So quite a lot of the immune system remains in these mice. So that environment means that the nude mouse is generally not going to be a permissive host for the engraftment of primary tissues. So it can be great, very useful for engraftment of immortalized cell lines, cells or tumor models that can be grown in culture, like our example of the MCF7s on the previous slide. But generally they are not the appropriate host strain for engraftment of primary tissues. So that enters in the highly immunodeficient NOD-SCID gamma or NSG mouse. And so we see the nomenclature and the official Jack stock number for that strain here. And so let me walk you through a bit about the NSG mouse and the phenotypes that are associated with it. So the non-obese diabetic background, or the NOD background, the inbred background that this strain is congenic upon, actually has its own unique immune system phenotype. So the NOD background has the absence of hemolytic complement. These mice are unable to successfully form hemolytic complement. They have reduced dendritic and NK cell function. They have diminished or defective macrophage function. And they have a more human-like SERP alpha allele on macrophages. So better enabling engraftment of primary human tissues because of that NOD background. Additionally, they have the spontaneous PRKDC SCID mutation. So the PRKDC SCID mutation, severe combined immunodeficiency mutation, impairs the ability of double-stranded DNA breaks to be repaired in the process of BDJ recombination. And so the functional effect of that mutation is preventing the functional development of mature T and B cells. So now we're combining defects in innate immunity, defects in adaptive immunity. And then lastly, what we have here is the interleukin-2 receptor gamma knockout mutation. So a full knockout allele that eliminates signaling from 6 distinct interleukins and very importantly, blocks natural killer cell development. And so we take all of this and put it together in the context of our pretty rudimentary schematic of immunity. And what we can see, if you contrast that in your mind's eye to what we were looking at in our nude mouse, is that the NSG mouse itself is a highly immunodeficient host strain, much more so than the nude mouse. And so this extreme level of immunodeficiency ultimately makes them a more permissive host for the engraftment of primary human tissues. There are a number of applications and benefits here. You may have come across the concept of leakiness of the SCID mutation, perhaps due to compensatory DNA repairing mechanisms that can occur to various degrees in different inbred backgrounds that carry that SCID allele. Well, for the NSG, there's no known associated SCID leakiness. Additionally, these mice have a longer lifespan than non-skin mice. Non-skin mice are known to develop thymic lymphoma at a fairly early onset. And this is something that is not seen commonly in NSG mice. They do have a long lifespan. And they have enhanced primary human tumor and hematopoietic stem cell engraftment due to that unique SERP alpha allele and their overall profound immunodeficiency. Now, because of the phenotype of the host strain itself, there are a number of important considerations that you must account for if you choose to work with NSG or NSG variant strains. They are extremely immunodeficient, and so they require a very careful housing and husbandry practices and careful planning within your facility to be able to use them successfully. Now, as I mentioned in an earlier slide describing the PRKDC skin mutation, its effect is the inability of double-stranded DNA breaks to be repaired. And that works to result in immunodeficiency in terms of VDJ recombination, but we're not able to be as selective about that per se. So because of the effect of that mutation, mice with the skin mutation are more sensitive to radiation and genotoxic drugs because of their inability to repair double-stranded DNA breaks. So there is an alternative to SCID if that is something that is of importance to you experimentally. And that would be in the form of the RAG mutations, which would have a different effect than these SCID alleles. So if that is something that's important to you, don't hesitate to reach out to us, and we can certainly direct you to some additional information on that particular topic. So again, a wide variety of applications and benefits to the NSG mice, but clearly some important considerations that must be taken into account if you choose to work with them. Okay, so I have another poll question here for you, and let me kind of walk you through it because there might be some nuances to the question that need some explanation. And so what I'd like to have you weigh in on here is which of these mouse strains you think is more likely to engraft primary human cells. So let me walk you through a little bit here. So we see in the first example, the CBY-SM SCID. The common name would be the BALB-C SCID. So you're probably familiar, but may not be, that BALB-C would be another inbred mouse strain, an immunocompetent mouse strain. And this would be a strain carrying that SCID allele. And then here we have the NOD, non-obese diabetic, carrying that SCID allele, NOD SCID. And then here we have our NSG, the NOD background, the SCID allele, and the interleukin-2 receptor gamma knockout allele. So my question here is, which of these strains do you think is more likely to engraft primary human cells? If you could weigh in there on our poll question, and then Dolores, when you've got enough responses, feel free to close that and broadcast those results. Great. You are all right on the money. Indeed, the NSG mouse, due to its profound immunodeficiency, more so than either of these other strains, is more likely to engraft primary human cells. So the thing that I want to illustrate, and I think we successfully have gotten there based upon your responses, is that looking at the strain in a holistic context, genetic background, and the mutations that it carries is really critical to identify the strain that's going to be most appropriate for your experimental applications, including engraftment of PDX tissue. Okay, so now that we have a good fundamental understanding of the NSG mouse as the host strain for engraftment of PDX, let's go ahead and dive in to our discussion of how PDX models are used for predictive and preclinical research. So let me walk you through a little bit about the JAX patient-derived xenograft program. So here we see kind of a schematic of how this would play out. We would begin by obtaining human tumor tissue direct from the patient, direct from the clinic. That would then come into our laboratory and then get engrafted into the initial NSG mouse, which we would term the P0 mouse. And then we would wait see if that tumor is able to be established and grow. And then if it does, that tissue is harvested from that initial P0 mouse. Some of it is frozen down in our cryobank. Some of it would be used for gene expression analysis, copy number variant analysis, and then some of it would be engrafted into another batch of NSG mice termed the P1 or passage 1 mice to expand that tumor tissue out further. And then we would be able to archive that PDX tissue in our cryobank. So there are a number of pieces of model information, characterization information that is obtained as these unique models are established. So we are able to gather clinical information from the patient, tumor type, grade, and any markers if there are known, some summarized patient treatment history that may come in with that sample from the clinic. We're able to perform histology and then also gene expression analysis, copy number variant analysis, variant analysis using the JAX Cancer Treatment Panel or CTP for mutation analysis as well. So some pretty extensive characterization that goes into these models as they are established and banked. So once that initial phase is done, what we are then able to accomplish for researchers internally, as well as those spread further afield customers that we're working with, is we are able to provide access to this PDX tissue. And so what we'll do is take a fragment that has been validated to grow in NSG mice and establish PDX from our cryobank and graft it into a panel of donor mice. And so the purpose of these donor mice would be really to expand the tissue out. So that tissue gets expanded out in the mouse host, which would then get engrafted into a cohort of mice, a larger number of mice that you could then enroll on study. So one thing that I want to point out here is there's nothing biologically different between the donor mouse and the cohort mouse. It's just a matter of we need to use the mouse host to expand the amount of tissue available to engraft into that cohort for use on study. So you can keep track, as you probably are here, in the number of passages that we're referring to. So a P0 as the initial, or passage 1 as the next passage in the mouse, 2, 3, and so forth. So we want to keep track of that, as you know, you want to be aware of what passage your tissue is and monitor for any changes in marker status. Make sure that your marker is still present across passages. So that's kind of an overview of what the process would look like for the JAX PDX bank. And so here is a representation of the models that are available from the JAX patient-derived xenograft bank. So we have over 400 clinically relevant PDX tumors, and they are at a high degree of characterization in terms of genomic, molecular characterization, histology, some standard of care information associated with them as well. And we'll look at that in subsequent slides in a little bit more detail here. So as you see, we have a well-established lung and colon tumor collection, as well as an extensive triple negative and ER positive breast tumor model collection. Of note, we do also have 2 prostate models in the bank, which are historically hard to grow. So that is something that is unique about this collection, as well as identified pediatric tumor models in support of the RACE Act as well. So let's look at a little bit more information about, you know, the benefit that you can glean or gather from engrafting these tissues in NSG mice. And so here we have some images taken from a 2008 publication with the PubMed ID link down below. So when you get access to these slides, you can absolutely grab that paper and drill down into more detail. And so what we have is histology at the H&E level from a patient lung tumor fragment. And then we see engraftment into either the NSG in the top panel of images or the BALB/C SCID. So very different mouse strains, very different in their level of immunodeficiency. So what we see is that at the H&E level, engraftment in the NSG allows for much more faithful recapitulation of the histology that you would see in the patient versus what you would see when you engraft in the biopsy skin. When we look at IHC for human cells by staining for anti-HLA, we see many more human cells in that tumor fragment engrafted in NSG versus what you would see in the BALB skin. And in parallel, when we look for mouse blood cells, staining for mouse CD45, many more mouse blood cells infiltrating in that tumor than you would see in the NSG. So more faithful recapitulation of the patient tumor when engrafted in the NSG mouse. And we can take that out a little bit further, looking in the top panel at the patient tumor histologically at the H&E level, and then looking at staining for cytokeratin, and then a proliferation index looking at Ki67. And here we see the engrafted tumor in the NSG. Again, more faithful histologic recapitulation, similar staining in terms of cytokeratins, similar proliferation index. So together, these two slides really support the idea that the severe reduction in the immunity of the host NSG mouse really permits more faithful modeling of that human tumor. And so here's an example that I'd like to illustrate a little bit further about colon PDX structure fidelity as an example over passages in the NSG mouse. So here we see an H&E of the patient tumor. Here we see the H&E of engraftment into that P0 or initial mouse, where we can appreciate the retention of these crypt-like structures. And what we also see is the maintenance of that when we engraft into that P1 mouse as well. And then in a separate example, we can see colon and adenocarcinoma that was metastatic to lung as the sample that was taken from the patient and engrafted here into the NSG mouse. So we can see maintenance across maintenance of the architecture of the tissue when initially engrafted into the mouse as well. So let's go ahead and have a look at some experimental data that can show you what some of these experiments might look like when we're looking at treatment response. So here we see an example of a bladder tumor model that was engrafted in NSG mice and then treated with cisplatin. And so we do indeed see a nice example of a response of that PDX tumor to cisplatin over time versus what you see in the saline treated control. So let's go ahead and drill down a little bit further and look at a spider plot. So here we see tumor growth curves for the individual mice from what you would see in this aggregate graph here. And the thing that I'd like to highlight is the amount of variability that you can expect to see. So one of the known features of PDX models is heterogeneity, right? Heterogeneity that can be a feature of working with diverse set of human tissues. So this is something to consider is that what you may need to account for in your experimental design would be sample size to allow for sensitivity, to allow you to detect a difference if it is going to exist. So what I would say is that this variability may in fact be more reflective of human tumors and can also potentially facilitate studies of drug resistance as well. So just an important aspect to consider when you're looking at these models. And so that's variability within a model. We can also appreciate that there is variability between models. So we can see here that in our first example, we were looking at TM00016 as our model ID, where we see a growth curve looking like this. And then if we look at a similar experiment, again, treatment with cisplatin in another bladder tumor model, we see pretty significant differences in terms of the overall growth kinetics of the model. So, you should consider that tumor growth rate and response to therapy will vary by model. And so you may consider screening more than one tumor model per cancer type, depending on your model selection indications. So another advantage, as you may appreciate kind of from the differences that can exist both within and between models, is that experiments can be designed to examine test article response in models with different genomic profiles. So what I have shown here in this schematic, again, would be our process to establish those PDX tumors. And then we can take the characterization information in terms of molecular assessment to use that to inform a panel of models for different treatment regimens based upon their molecular types. So here we see looking specifically at the EGFR gene, looking for non-annotated variants in EGFR. And then activating mutations or different mutations in EGFR, L858R, L858R, and T790M that are known to be associated with different resistances or models that have resistances. And so then you can apply these different pieces of information about the molecular assessment of the models to your different treatment regimens. So genomic characterization in response to standard of care therapies that we have annotated to individual model detail pages, which I'll show you at the end of the talk, can be combined to enable and facilitate a more robust experimental design as you're conceptualizing your studies. So I'd also like to spend some time discussing marker status in breast tumor models. So this is a significant achievement in that breast tumor PDX engrafted in NSG mice are able to support the retention of estrogen receptor positivity status. And so here we see in this example, TM00386, we can see IHC for estrogen receptor in the patient tumor. And then we see that retained in the passage 5 PDX tumor. We also see retention of an organoid growth pattern. An important consideration here is that supplemental estradiol is used to support estrogen receptor positive tumor growth in these mice, and that NSG mice are relatively resistant to estradiol-induced bladder toxicity versus other host strains. So a very important feature for the modeling of hormone receptor positive breast tumors. And we can see that borne out a little bit further when we look at further histology. Again, in that 386 model, looking at that organoid pattern in the patient, as well as the initial passage in the mouse and into the P1 mouse as a follow-up there. Okay, so we kind of touched on this looking at our EGFR example. But one thing that can be particularly powerful is to be able to mine the molecular characterization information to predict a therapeutic response. So here we have a non-small cell lung carcinoma, TM00186. We see histology. And what we know from a gene expression standpoint is that this model carries a variant in KRAS, the G12C variant here. There is no EGFR variant annotated, and the model is characterized as ERK1 low. So that should mean a predicted sensitivity to cisplatin due to impaired DNA repair mechanisms. The lack of annotation of an EGFR variant would predict resistance to erlotinib as an EGFR therapeutic. So these would be the predictive responses based upon this gene expression profile. And so then what we're able to do is take that to the experimental phase and see if that is indeed borne out. And so here we see in the TM00186 model that I described in the previous slide, treatment to cisplatin and erlotinib. We see tumor volume here, days here, and as you would expect, we see the vehicle is perfectly happy to grow. We do not see a response in erlotinib, in the erlotinib treated group, as would be predicted based upon no annotated EGFR variant. And we do see a response to cisplatin, as would be predicted based upon the ERK1 low status. So we can look at that in another example here. So what we're looking at in model TM00199 is what you would see, or what you would see at the level of the patient. So let me orient you to this particular visual. The area under the curve is representative of patient tumor burden. So this is in the patient that the TM00199 model was established from. And so what we see is the patient was initially responsive to erlotinib therapy, and this is in a tumor that is known to carry the L858R mutation in EGFR, so predicted, and then we see response to erlotinib therapy. And then ultimately what we see is a lack of response over time to erlotinib. So we see progression of disease on erlotinib therapy. And so once this resistance to erlotinib was established, that is when the sample was taken and the PDX model was established. And so we see progression on erlotinib therapy, and then ultimately we see a change in treatment to a combination of afatinib and cetuximab, and there we see regression of the tumor tissue. And so this is what you would see at the level of the patient. And so then we're able to probe that in the same way when we look at the PDX model and graft it into the mouse. And so again, I'd like to highlight that the PDX sample was taken at the time the patient was progressing on erlotinib therapy. And so let's have a look at the growth curve in that particular context. So here again, we have tumor volume on our Y, days post-treatment on our X, and this is in the established PDX model. We see growth in our vehicle-treated group. And then what we see, as was mirrored in the patient, we see progression of tumor growth on erlotinib therapy. And we see suppression of tumor growth with afatinib alone. And then we see more marked suppression of tumor growth with cetuximab alone, or the combination of cetuximab and afatinib. So the treatment response fidelity in this particular example is maintained between the patient and the PASSAGE PDX tumor model. So if we put these last two examples into broader context, that perhaps by combining PDX gene expression data and experimental efficacy data, we can potentially be able to predict treatments that will be most efficacious based upon individual tumor expression profiles. So pretty powerful system that we're able to work in there. Okay, so toward the last part of this talk, what I'd like to do is just orient you briefly to immunologically humanized models for immuno-oncology applications. And as I mentioned at the start of the talk, we have a number of other webinar presentations and resources that go into much more detail on these model systems, because they are quite nuanced and quite complex. So, we definitely want to equip you with information on those topics as is of interest to you. So please reach out to us and we can certainly get you more information there. So we'll just do a really brief overview here. And so the overview that I'd like to share with you is in using a CD34 positive hematopoietic stem cell humanized NSG mouse in this particular example. There are a number of methods of immunological humanization. CD34 engraftment is, HSC engraftment is one. Also, peripheral blood mononuclear cell engraftment is another. Both of those are done regularly and readily here at JAX to be able to support your needs. But here's an example or a schematic of how you would go about developing a oncohu mouse or a immunologically humanized tumor-bearing mouse for use in some of these studies. So we have sublethal irradiation as a means of preconditioning prior to HSC engraftment. So then we have hematopoietic stem cell engraftment by tail vein injection to result in our immunologically humanized NSG mouse. Once that mouse is validated and confirmed as humanized by looking at human CD45 staining in the peripheral blood, you can co-engraft a PDX tumor or cell line xenograft depending on your needs. And then ultimately what we're able to do here is study a human tumor, PDX, or cell line xenograft in the context of a human-like immune system in the mouse and enroll those on efficacy studies to be able to study that in a particular experimental context. And so one of the initial questions that needed to be addressed when these models were initially established was, whether or not there was going to be a difference in the PDX tumor growth kinetics in those immunologically humanized mice, humanized by CD34 positive hematopoietic stem cell engraftment, versus the non-humanized host. And so here we see 3 different PDX tumor models engrafted in the humanized NSG mice shown in the pink line or non-humanized NSG mice shown in the green line. An important point to address here is that these are PDX tumors that were already established to grow in NSG mice. So it's an established PDX model that was then tested for growth in a humanized NSG mouse. And a note on HLA matching between the CD34 positive hematopoietic stem cell donors is that partial or full HLA mismatch may be tolerated here. But what we see is that in all of these examples, the growth kinetics of the non-humanized host with that tumor on board and the humanized host with that tumor on board were relatively similar for these 3 different models. What I would highlight here is that PDX growth kinetics should be tested for each model in the humanized system before enrolling on study. And then due to the variability in co-engraftment and therapeutic response, several different CD34 positive hematopoietic stem cell donors should be considered per experiment. We know there are donor specific, donor dependent effects on treatment response. So you want to spread that variability out by using multiple CD34 donors across all arms of your experiments. So here we can see an example of what some of those experimental data might look like when using a humanized mouse with a PDX tumor on board in an efficacy study example. So humanized mice can respond to checkpoint inhibitors, and this is going to be in a CD34 positive HSC donor dependent fashion. So again, really important to use multiple CD34 donors if you're planning to do checkpoint inhibition studies, really studies in general using this system. So the way this works, if you're not familiar, is that Keytruda, our example in this particular experiment, binds and blocks PD-1 on T cells and prevents its interaction with PD-L1 on tumor cells, ultimately removing a tumor's inhibitory signal on T cells, enabling that T cell to act and kill that tumor cell. And so what we see in our example Again, tumor volume on our y-axis, treatment on our x, a nice growth curve in our vehicle-treated group. We use cisplatin as a standard of care therapeutic that does indeed cause regression, suppression in tumor growth, and we are also able to see suppression in tumor growth in the Keytruda treated group as well. And important to note, the response in this scenario is likely allogeneic, as the CD34 positive HSC donor and the PDX donor are different. So this study really does demonstrate that the OncoHu model can be used to test efficacy of immunomodulatory therapeutics in this context. All right, so one final poll question that I have for you. Which of these would you predict to be useful for cancer immunotherapy research? So Dolores, if you could open up that poll question, that would be great. Right. Great, great. Yes, absolutely. Most of you have selected an NSG mouse with a PDX tumor and human hematopoietic stem cells engrafted. Absolutely, based upon the example that we looked at here. Let's talk through some of the other examples just as a means of reviewing. So our NSG mouse with a PDX tumor on board. Remember, the NSG mouse is a highly immunodeficient host. So we're not going to be able to study a human-like immune system unless we co-engraft that here. So a nude mouse with a PDX plus human hematopoietic stem cells. I would say the challenge here would be to get that system to be robustly established, because as you'll remember from our earlier slide, the nude mouse, the primary immunodeficiency is at the level of the T cell, making it likely not the appropriate model for PDX or HSC engraftment. And then our first example, so we're looking at a syngeneic cancer cell line, the B16F10 melanoma cell line, syngeneic to a C57 black 6J mouse. Indeed, this could be a useful model for immunotherapy because the black 6J mouse, 57 black 6J, is an immunocompetent host with a matched syngeneic cancer cell line. The caveat here is that this would be a mouse tumor in a mouse immune system. So there would need to be ability of the test article to interact specifically with the mouse as distinct from being targeted toward humans specifically. So some limitations there, but these are an absolutely a viable option depending on the test article that would be employed. So great. Thank you for weighing in there. Appreciate it. Okay, so that takes us toward the last bit of presentation material that I'd like to highlight. And that is in regard to one of my favorite resources, and it exists within the Mouse Genome Informatics database as a subset here, and it is called the Mouse Models of Human Cancer database. And so this is going to be the place to go to begin your search of the JAX PDX tumor collection to identify models that may work for your particular experiments. So let me walk you through how some of that might work here. Okay, so here we see some screen grabs of what that database looks like. And what you can do by accessing our PDX search form from that MMHCDB website would be to search by a number of different parameters. You can search by tumor ID, the ID that is unique to that PDX model if you know it. You can also perform searches by primary tumor site. You can perform searches based upon diagnosis that would have come in with that cancer from the clinic when the PDX was established. You can perform searches based upon gene variants that are annotated based upon the molecular data for a particular model. And you can also look at dosing study information. So some of the models, many of the models now that we have in the collection at JAX have standard of care dosing study data that are annotated to the individual model detail pages. And so as you can kind of see in our screen grab here, you can limit your search results to those that have dosing study information to be able to look at that as well. So a number of different parameters that you can perform searches by. What I will illustrate here is that the genes that you can search and visualize on the individual model detail pages are going to be a subset. So they're going to be genes that are part of the JAX cancer treatment profile, the CTP. So it's not going to be all genes reflected. So if your gene of interest is not present, from a data standpoint, what you would need to do is reach out to us at Mice Tech, and then we can work with our PDX informatics group to access that information for you, and then we can certainly share it with you. So that's one thing that I want to highlight for you here. Okay, so here we can see an example of what a search would look like. So if we were to search on triple negative breast cancer, so that would be a tag at the level of the patient. So patients that were diagnosed with triple negative breast cancer, and then PDX models were established from tissue from those patients. And what we can see here is that there are 30 matching PDX models in the JAX bank that are annotated as TNBC or from TNBC diagnosed patients. And then we can see what additional data may be available for those models. So dosing studies, histology, et cetera. So let's go ahead and have a deeper look at what we'd see on a model detail page. So here we see PDX model TM00096. We see some kind of overview information for the model, including summary, clinical diagnosis, etc. And then if we look down further, what we can expand out to see would be a variant summary. And I'm going to show you a little bit more about that. We can also see gene expression information that would be annotated, copy number variants, etc. So here we see an example of the gene expression information that you can expect. So we see a waterfall plot of our genes here, and we can look at the samples that these were taken from. So an RNA-seq, patient 0, or a primary P0 mouse, I should say, is where that would come from there. And so we can see an example of what those data would look like. And then this is particularly powerful, and I'd like to spend some time on this. The variant summary information that is accessible on the individual model detailed pages. It crosstalks with the JAX clinical knowledge base, the CKB, which allows you to access really a lot of predictive information about that particular variant that is annotated for the model. So the CKB is a dynamic digital resource for interpreting complex cancer genomic profiles in the context of preclinical and clinical trial response data. So if you click on a particular gene variant, seeing that hyperlink there, you can be taken out to a CKB molecular profile page and see associated information with that. So I highlight this as a particularly powerful way to drill down into literature associated, data associated with variants annotated for these particular models. So here we can see an example of the standard of care dosing study information that you may be able to access for models in the MMHC-DB. So we can see agents that were used to treat that tumor with, mice carrying that tumor with, as an example, several standard of care drugs. You can see an overview of study design, dosing information, growth curves, as well as other representations of the data, including a spider plot to look at variability, which can be particularly helpful, plots of tumor growth inhibition. So really a valuable resource to be able to understand how different models have been characterized and make decisions about their suitability for your particular experimental applications. All right, so it looks like we are right on target to get toward our Q&A section, so this is great. Okay, so in summary, I hope the data that I've shared, the information that I shared, illustrates to you that PDX models can more closely recapitulate clinical observations than previous mouse models. And this includes gene expression, tumor structure, as well as treatment response. I hope I've also shown to you that low passage PDX samples can retain the heterogeneity as well as the fidelity of the human cancers, and that we do have significant characterization data available to help guide your model selection decisions, and that you can take these tumors that meet your needs from a molecular signature or your particular selection criteria and pair them with immunologically humanized mice for immuno-oncology applications and efficacy studies. And these are things that scientists at JAX are working on daily, both from a research standpoint and also from a solutions and services standpoint, that we are able to work with you to run customized efficacy studies with these model systems to meet your needs. And again, so here you can see an example of the number of services and solutions that we can provide to you in the way of model access, as well as preclinical solutions like our in vivo oncology efficacy studies. So if that is something that is of interest to you and would help provide solutions for your program, do reach out to us and we will absolutely be happy to have a further conversation with you about that.
One question that came up is why are prostate cancer models so difficult to model with this PDX strategy?
Yeah, it's a really good question, and I don't have a great answer for the why. I can tell you that it's pretty repeatable that they are difficult to establish. They are difficult to establish and to be able to use, you know, from a model establishment standpoint. And so at JAXA, we do have 2 prostate tumor models in our bank that are available for use. And what I would highlight also is that one of them is part of what we call our PDX Live tumor model selection. And so what that means is it's a selection of PDX models that we commit to maintaining live on the shelf, which does decrease the lead time and broadens accessibility to those resources. So if you are interested in using a prostate tumor model, you know, that's going to be a good thing to keep in mind. As far as the why prostate tumor models are a difficult type of PDX to engraft, you know, it's an important and interesting question, I think, and does also highlight that, you know, even though we've made significant strides in terms of using the NSG as a host strain for the engraftment and establishment of PDX models, there is still room to be able to make improvements, especially in the establishment of these harder to grow PDX models. So that is certainly something that researchers at JAX are committed to doing as well. So it's an iterative process to be able to continue to support these harder to establish, harder to grow models.
Absolutely, absolutely. And this brings me to the next question. You explained very nicely why and what compartments are non-functional in NSG mice that make them severely immune deficient. So the next question is, because they are so immune deficient, how do we house them? How do you recommend people to house them? Because they are, you know, so immune deficient.
Absolutely. It's a crucial consideration. And a question that in technical information services, we talk to folks about regularly. So, sterility and barriers associated with the facility are absolutely key. Individually ventilated cages, and making sure that feed and bedding and caging and water, really anything that's coming into contact with the animals or their environment is sterile. Being careful about co-housing in the same space with immunocompetent animals, limiting traffic flow to that particular space is going to be key. Working really, it's an ongoing conversation that needs to be had with your facility as well as your veterinary team to understand, what are the practices that can be put into place that are going to best support a strain that is as immunodeficient as this. The other thing that I would highlight, particularly in the context of our discussion around PDX models, is that these mice do have human tissue on board when they are engrafted with a PDX tumor. And so working with your facility, to understand, what's appropriate as far as barriers that need to be maintained for handling human tissue in a mouse is really key. At JAX, we're working with these at a ABSL2 level because they have human tissue on board. So that's something really important to address before you bring models like this into your facility. It's a really key question.
Absolutely. And the other problem that some researchers encounter, and they call us for tips on how to troubleshoot that, is the fact that you do need to test the cell lines or test the tumors for pathogens. Otherwise, these mice are very susceptible to get infections from the tissues that you're engrafted. And that, of course, that applies also to the humanized cells that you would use to humanize the mice as well if you're doing immuno-oncology. So that's a critical thing too. And we don't have time to explain all the pathogen tests that we do here. But if you're interested in knowing what we test for, I encourage everybody to email us at micetech@jax.org, and we'll be happy to give you more information.
Absolutely. Great point.
All right. So last question, and if you could briefly say, one of our listeners is asking, how easy is it to generate humanized NSG mice in-house with patients' hematopoietic stem cells? And I know that we have a whole webinar on this topic, but if you could just say briefly, in our experience, I mean, I don't think it's something easy to do, but I'll let you expand on that.
Sure. I mean, I think that you need to think about all of the steps that would need to occur in the process, right? Optimizing technique is one piece of this as well, but then you also need to be able to optimize a validation program to be able to validate your engraftment. Timeline is also, a key consideration here. We didn't talk about this in great detail, but in the context of CD34 positive hematopoietic stem cell humanization, these take some time. So, after you engraft at 3 weeks of age or so, you're going to wait for multi-lineage differentiation and establishment of that human-like immune system to occur. So, and we're talking about, 12 weeks or so of waiting before you would even do your validation experiments to confirm engraftment. So, it's a resource intensive and a time intensive, as well as a very precise technical technique that must be optimized to be able to get this platform up and running. So with all of that in mind, it's going to depend on your needs. It's going to depend on the goals of your program. It's going to depend on, what your specific experimental requirements are and what is going to be the best fit for you. But what I would say is, we at JAX have tried to be able to fill this need that we know to be really important for folks to have access to this resource. And so we are readily humanizing CD34 humanizing mice and then PBMC humanizing on demand different NSG strains to make them available really off the shelf for folks to be able to access without having to wait for engraftment to occur and without having to do validation checks on your own. We're able to do that before we ship the mice to you to make ease of accessibility much more straightforward. So if that's something that is of interest to you, if you'd like to know more, please reach out to us and well, we'll be glad to talk with you in more detail. It's a really good question, an important one, and I hope that kind of provides some context there. I can't tell you what's going to be the best fit for individual users or individual needs, but I would say it's a complicated and nuanced model, and we want to be able to best support your use of that. And one of the ways that we can do that is by making them readily available off the shelf for you to use.
Very good, thank you, Grace. And the only thing that I would add to that is that we do have large inventories of already humanized NSG mice as well as humanized NSG variants. So they're ready to go, validated, ready to use. And then to answer the viewer's question, like if they really need to use hematopoietic stem cells from patients, we could do that as a custom order as well. So sometimes you know, for your own project, you really need to use specific cells from specific patients. So we are able to do that as a custom order too. Of course, it will take time because you have to let them mature and all of that, but that is something that we are also able to do. As far as how stable they are, in our experience, I mean, the engraftment is it can go on for at least 12 months. So it is very stable on the NSG host. So Grace, any final thoughts before we go?
Just a note of thanks to all of you for spending some time with us. I hope it was of value. And if there are specific questions or if you want to talk about your particular program, please reach out to us. And we're always glad to speak in more detail.
Thank you so much, Grace, for this wonderful presentation. And to our viewers, before you go, please fill out the survey that will pop on your browser when you leave, and you can tell us how we did today and what other topics you'd like to hear from us in the future. So thank you, everybody, again, for participating. Thank you for us answering the poll questions, and you have a wonderful day.